Hello students, in this module I'm going to talk about the process of translation. Look, listen and learn. Let us again recollect the central dogma of protein synthesis. Remember this RNA molecule you just made by transcription? You guessed it, this mRNA molecule will now get translated into a protein. And what is a protein? Nothing but long chain of amino acids. Is that okay? And this process is called as translation. Now, remember where translation will occur. Translation will occur in a eukaryotic cell in the cytoplasm. On what part will translation occur? Translation will occur on this special structure called as the ribosome. Now on the ribosome you have this mRNA which is going to get translated into the amino acid sequence which is protein. Is that okay? Moving on. Let us understand how it happens in short. Okay, so the process of translation basically involves the mRNA strand, right? And this mRNA strand basically is made up of what? It is made up of codon. And recollect, a codon specifies an amino acid. And you can see how codons are specifying amino acids. Is that okay? So how would I define translation? I can say that translation is a process, right, where the sequence of codons on the mRNA are read or decoded to basically give you amino acids which are joined to form the polypeptide chain which we call as the protein. Is that okay? And that is translation for you. Now, let us see what are the requirements for translation. Translation first requires molecule of DNA. Do not forget, it is DNA which will give you RNA and then the RNA will give you the protein. Is that okay? Then we also require 20 types of amino acids. We don't know which amino acid will be required when. Then we require non-genetic RNAs and all three of them, mRNA, rRNA and tRNA, each one doing their specific role in the process of translation. Translation happens where? You guessed it right, on the ribosomes. So we require ribosomes. And then for the purpose of translation, we require many enzymes or factors. And finally, the process requires a lot of energy. And the two sources of energy that we use for translation are ATP, and whenever ATP is not available, we can also use GTP. Is that okay? So these are all the requirements for the process of translation. Is that okay? Moving on, let us understand the process of translation. I've just shown you one diagram where you can see the entire process of translation happening. But the main process of translation can be divided into two basic steps. And the two steps are as follows. The first step basically involves activation of amino acids. What we are doing here is adding the amino acid onto the tRNA. And the second step is bringing these amino acids together and nothing but synthesizing or forming the polypeptide chain. So these are the two basic steps of translation. Step 1 is activation of amino acid and step 2 is formation of the polypeptide chain. Moving on, let us see how the activation of amino acid takes place. For the activation of amino acid, we require a very important enzyme, and the enzyme is amino acyl tRNA synthetase. Let us see how this enzyme looks like. There you go. What I've shown you here is a molecule of the enzyme called as amino acyl tRNA synthetase. Right? What will it do? It will first bind to the amino acid. So let's bring in the amino acid. What you see up there is an amino acid. And now let's put this amino acid on the enzyme. Where will you put it? You guessed it, on that site which is called as the amino acid binding site. Shall we do it? There you go. We just added the amino acid on the enzyme and we got the activated amino acid. Is that okay? Now what we'll do with this amino acid? We have to take this amino acid with the help of that enzyme and put it on the tRNA, right? Look at the other side there. What is that site called? That site is basically called as the tRNA binding site. If you remember, on the tRNA, there was a DHU loop where this enzyme will bind. Is that okay? So let's go ahead and bind it. So this is my tRNA, right? And there you go. The 3' end. Remember 3' end was called as the amino acid acceptor end. Let's go ahead and take that amino acid and put it on the 3' end. And let us see how. So this enzyme will come forth, right? And the presence of a molecule of ATP will take that amino acid and put it on the 3' end of the tRNA. This step is called as the charging of tRNA. It is also called as the amino acylation of tRNA. And the final complex you see here is then called as the amino acyl tRNA complex. Is that okay? And this step is called as the activation of amino acid and forming of the amino acyl tRNA complex. Is that okay? So in this module, I introduced you to the process of translation. In the next module, I will show you how exactly is the protein formed by simply talking about the formation of the polypeptide chain. Thank you and take care.